So my name is Dr. Brooke Johnson. I'm going to be talking to you today about some of the work I've been doing on a Proterozoic and a separate Archean succession. It appears that we are sans wolf. So I'm going to be introducing myself. So I did my undergrad at Birkbeck College, University of London, and then did my PhD with Nick Tosca and et al. At Oxford, and that was on the Greater MacArthur Basin. I'm going to be talking about later. And now I'm in Liège working with Emmanuelle Javot in her lab, looking at some Archean material. So first of all, I'm just going to give a bit of a background about why we're looking at this kind of stuff. Then I'll tell you about the Greater MacArthur Basin, which is a Proterozoic basin that I've been working on, smack bang in the middle of the mid-Proterozoic. And then I'm going to talk about some of the unusual putative microbial structures that we found in the deeper water clastic settings of this basin. And then at the end, I'm going to talk to you about the Moody's group, which is the Paleoarchean material I've been working on. This is really exciting stuff because it's freshly drilled material that's unweathered, and it's a really beautiful material. So just a quick note is that you'll notice I use lots of different colours because I'm dyslexic, and having all of the different colours helps me keep track of where I am. If it's relevant to the data, I'll let you know. So I haven't done any of this alone. A lot of people have helped me, and for the Proterozoic material, this is just a wee section of the people that have helped me out, specifically Emmanuelle and Yannick in Liège, and Alex, and then Rosalie, Nick, Stuart, Kat, and Amber from Geoscience Australia, Oxford, and Cambridge. So we want to try and find life on places like Mars, and a good way to go about that, or try to figure out where you might find things on that were fossilized on early Mars, is to think about early Earth, because Noachian Mars is thought to have been similar to Archean and maybe even up to mid-Proterozoic pre-cryogenian Earth. And so we're thinking about places like the crater lakes on Mars where we find all of these clays, and where the probes have been landing recently. So that's one of the reasons that we're looking at this material. So we know that fine-grained clastic sedimentary rocks are an archive of fossils, and particularly organic material on Earth, but we don't really understand fully why that is, or why my slides keep jumping on their own. So some people think it's the antibacterial properties. Some people think that the clays are templating soft tissue. And other people have shown that rather than the specific clay chemistry, what's happening is that the clays are reducing permeability so that things like metabolites and bacteria can't permeate through the sediment and destroy the organic matter. So to look at this, we looked at material that I'd collected from the Greater MacArthur Basin, and this is a cratonic basin. It's analogous to one of the Martian Greater Lakes in a way, in that it was deposited by a big river system in an arid environment, very low oxygen, and very low oxygen, and it was draining a basaltic hinterland. So it's right bang in the middle of the mid-Proterozoic, and we've got all of these different both contemporaneous, geographically and temporally, units that are really well preserved because they've been buried, and it's all from drill core material. So we collected a lot of samples and ended up logging many kilometers of core, from which we've extracted a lot of data, particularly many thousands of fossils, which we've looked at for the preservation quality and other features. And then as well, we've done things like SEM and TEM and Raman to study these organic matter. So I'm quickly just going to talk about some of one of the techniques I use. It's a little less common, and that's macro photography. And I've been using this to look at structures on bedding planes. And so these are all very small fossils or structures, like those algae are hundreds of microns across. But by using macroscopic camera lenses, we can photograph them and get really high resolution images. We can combine that with traditional microscopy to really look at these things optically at levels we wouldn't normally be able to do that. So the way we do this is we set the sample up underneath the camera with the macro lens. That's just the one I use. There's lots of different types. And then we use an adjustable platform to raise and lower it. And so in this middle section here, you can see we've got the light green, which is the material that's in focus, and then we've got the darker green, which is the material that's out of focus. And you take lots of slices through from the sample, going from its highest point down to the lowest point. And that lets us, we then get to stack them together using a program called Helicon Focus, and then they build up into a full image of the actual object. And this is because the focal plane is so thin when you're using these macro lenses, it's microns thin. See, some of you are saying that you can't hear me properly. Is this any better? How about if I hold it up there? Okay. Okay, no problem. Right. So the first thing we're going to look at is these acritarchs. These are organic walled microfossils. And acritarch basically just means of uncertain affinity, uncertain origin. We don't know what they are. Ones like these ones here, they're probably eukaryotes because of the set of features that they have. They're relatively complex and generally too complex to be really considered to be prokaryotes. And that's the main mode of preservation that you get in the Greater MacArthur Basin. There's also rarer types of preservation, and such as these carbonaceous compressions that we see here. We're not really sure what they are. They're a bit too well structured and coherent to be and consistent in terms of the morphology to be just generic bits of microbial mat. But we're still trying to work them out at the moment. Okay, so we set about creating a framework to see how well preserved our fossils are. And at one end, with a mark of 0, you've got something like this kind of vaguely vesicle-shaped smear. It's made of organic matter. We can test that with Raman and show it's kerogen, but you can't really tell what it is. It was probably a fossil, but beyond that, you can't say for certain. And then you get increasingly better preservation. So the next one, we can see that was definitely a fossil. Can't tell what kind of fossil it was. It was just some kind of vesicle. The next one is clearly a vesicle. There's some internal structure, some external structure. And then as we get up to good and excellent preservation, we can see that there's very high resolution structures there. And on that one where we've got on the far right there with excellent preservation, the blue and green arrows are pointing to nanoscopic details that we can just about make out there. And we've been able to confirm those with SEM and TEM. So those are real actual structures that have been preserved on the outer surface of that particular kind of acritarch. So as well as looking at the preservation quality of the fossils and the organic matter, we also looked at how that compared to a lot of different factors within the basin. And so I've listed some of these here and put these together in this correlation matrix where blue is a positive correlation and red is a negative correlation that's statistically significant. And the more saturated the color, the more significant the correlation is. So there was something like total of 24,000 data points, and this is only like 10% of them shown there. So we're comparing preservation quality to primarily clear mineral abundance and species, but also things like the bulk types of minerals, acritarch species richness, boreal depth, where the where they were situated within the basin, and if there is microbial structures present within that sample or not. So first thing we looked at was total clay abundance versus preservation quality. And there's a negative correlation, but it's not statistically significant. We did find, though, that there was more of a correlation between preservation quality and different facies. And that's basically the depositional environment where the fossil was recovered from. And so we can see that here, that when you look at the different facies, we've got preservation quality increases and decreases independently of things like the clay abundance. Also found that there was no specific clay species that really matched with the preservation quality of the Acritarch fossils. So in one part of the basin, berthurine, for example, would be associated with poor preservation, but in a completely other part of the basin, berthurine would be related to and in association with excellent preservation. So it seemed more that the physical depositional environment was more of a factor in preservation than the actual clay chemistry or abundance. So what I've done here is I've tried to group these together by positive correlations and negative correlations on preservation quality, and then the length of the bar going from left to right shows you how long that particular factor on preservation quality was active. So the physical geographical position from where the sample was collected within the basin, that was something that was set when the basin was first starting to form, millions or even billions of years before deposition. Something like the systems tract of the sea level, where the sea level is going up and down, that's something that will affect preservation at the time of deposition, or, for example, and for a short while afterwards, but then it has no influence. And then things like position within the basin can affect preservation quality long afterwards, because if you're at the shallow end of the basin, you're more exposed to things like late stage weathering. Whereas if you're in the deeper part of the basin, you're not going to be affected by weathering, but you'll undergo greater diagenesis, and you are more likely to experience things like the intrusion of mafic sweets, which is one of the things that affected preservation in this particular basin. So it's, rather than being one particular smoking gun, so to speak, it's actually like a very complex network of interacting factors that go across both time and space that determine whether a particular fossil in a particular location within the basin is going to be well preserved or poorly preserved. So using this information, we tried to make a predictive model. So where in the basin would all of these positive correlations stack up, where we should get the best preservation? And so what we found was it's somewhere where you can have rapid seafloor orthogenesis, where there are a mechanism for coating grains and churning up the sediment to get fossils into the sediment, but also to stop the sediment being colonized by things like microbial mats, or to have hard grounds forming. Where you've got a balance between the amount of framework grains and the amount of clay being formed, and during the early stages of a sea level rise with moderate burial depth, and in a location where the burial was enough to keep it in the subsurface but not bury it too deeply. And so there was a location in the basin that matched all of those, and that was a unit called the Sherwin Ironstone. And it's this beautiful coated grain, ooidal and uncoidal ironstone, and the clay here is mostly berthurine and greenolite, with there's the grey stuff there is silica, and you get these beautiful sprout-like, they kind of look like Brussels sprouts to me, coated grains. And if we thin section them and look at them with a very high resolution optical microscope, you can see there's a lot of these interesting organic structures tied up within the layers of these coated grains, which means that they were picked up and incorporated into the grain at the time of deposition. So here are some shots of one of those grains that I've broken open and put under the SEM. And you can see as we get closer and closer, the grains aren't just made of a single solid layer for each layer. They're actually made of like overlapping sheaths or plates, kind of like a Brussels sprout would be. And that they have these little black carbonaceous structures in them that are about the right size and shape to be acritarchs and matched up with where we expected. We saw those things from thin section. So we did macerations, and what we found is a diverse suite of incredibly beautifully preserved acritarchs with the best preservation that we saw in the whole of the basin. So that one I showed you earlier with the nanoscopic details on its surface, that's from this particular unit. Okay. And so we're still working out what a lot of these things are, and that's, I can't really say much more about them at the moment, but there's a lot of really interesting forms in there. We also found these things, and if you look at the top left there, we've got what's called a Leofusimorph acritarch. That's basically just a rugby ball shaped acritarch. And as we go right and down, you can see that the organic matter is becoming increasingly worn away. And inside, what we can see is we've got this mineral material, and this is a mix of different types of clays, silica, and some phosphate. So what this says to us is that, or rather the way that we've interpreted, is that you're getting very, very rapid seafloor orthogenesis of these minerals. And it's so much so that we're actually getting them inside the acritarchs, and it's helping to preserve them with some level of three-dimensionality. So they're not completely, they're not 3D in the same way a dinosaur bone would be, but they're much thicker than a typical acritarch fossil usually is. And so they're much more resistant to things like diagenesis and all of the other processes that would destroy fossils. Okay, so how does this work out across the basin? Well, if you're in the middle, you've got a higher chance of entering the sediments, and you've got a higher chance of good long-term survival because of the rapid orthogenesis, just the right amount of reworking of the seabed. And then you're not going to be buried too deep or too shallow, so weathering can't get to you, but also things like extreme diagenesis, and then the effect of intrusive sweets. If you're in the shallow end of the basin, you're probably going to get destroyed by the higher energy levels. Anything that makes it into the sediment has a higher chance of being destroyed by things like heterotrophic reworking by bacteria. And then you've got a good chance of being destroyed by sedimentary erosion when there's a drop in sea level, and then weathering much later on during exhumation. And then at the other end of the basin, where sedimentation rate is very low, things are sitting around on the bottom of the sea for a long time before they're getting entrained in the sediment. So biomarkers from these deep water areas, they tend to have a lot of the biomarkers that have been broken down. I can't remember what they call them now, UCMs, but it's biomarkers that have sat around and they've been either heterotrophically reworked or oxidized, so you can't tell what organism they came from anymore. And as you'll see, there's a multiple reasons for this. One of them is that we see a lot of firm grounds and hard grounds, so the sedimentation rate is so slow that the sediment is turning to rock on the seabed. But there's another biological reason for that we're going to talk about in a moment. So yeah, primary clay chemistry doesn't appear to be that important. Rapid orthogenesis is more important, but only in the right place at the right time. It's really complex, which is, you know, what you'd expect. The Earth is a complex system, even back in the mid-Proterozoic. Okie dokie. Right, so we're going to talk about the microbially induced sedimentary structures, and there's just a quick definition for you if you're not familiar with what these are. So most people have heard of stromatolites, but we're in carbonate settings, but we're looking at things that were forming in clastic settings. In particular, we're looking at things that formed in relatively deep water, clastic settings as well. Okay, so we often find these in the Greater MacArthur Basin with in situ organic matter and material. I'm going to go through all the different types that we found. So here we've got these ones in different kinds of lighting, and you can see that you've got these little silvery ovoid structures. And in the low angle light, we've got these ridges that are radiating away from these ovoid structures, and these ovoid structures are organic matter, and with some mineralization around it as well. Got some beautiful SEM pictures, but I can't of these things, but I can't share them yet. So yeah, these are definitely fossils. So the most common type we get in the offshore areas are these wrinkly bedding plains where they've got this material on them that kind of looks like you've got draped cheesecloth or something over the sediment, and it's gone all wrinkled and scrunched up. And then if you look closely at this under, say, a microscope, you can see that there's all kinds of weird textures and filamentous material on it as well as these. You also get these unusual pellet chains, which is the purple arrows pointing to there. And when we look at these under the thin section on there on the right-hand side, you can see that you've got all of these dark layers that are full of organic matter and organic filaments and little spheres. And then you've got these lighter areas that are full of chert and different kinds of aluminous clay, particularly iron clays, and also these little phosphatic spheres as well. So again, we're seeing that we're getting very rapid orthogenesis. And in some cases, you can see where the organic elements, the filaments and whatnot, are growing over and trying to keep pace with or ahead of the actual material that's mineral material that's accumulating on there on the sea floor. And so those pellets, if you look on the thin section where the purple arrows are, that's cross sections through those pellets, and they form these kind of lensoid structures. And then when we get them lengthways, we can see there's also it forms a lens along the length of the little pellet. So we're not really sure what they are, but they're really abundant, and they appear to have been some kind of three-dimensional structure on the seabed because they interweave above and below the same microbial mat on the same bedding surface. So they were there at the time of deposition. And they're usually infilled by, like I said, iron clays or silica or aluminous silica. So the next one we have is these pellet chain dominated beds. And this is just more of these unusual chains of these pellets. I call them pellets because that's the best name I could come up with to describe them. So all of these names are descriptive rather than genetic. I'm not saying actually what this thing is. I'm just trying to describe what it looks like at the moment. And these are all interwoven with each other and growing over each other. And this particular kind of texture can form layers that are tens of centimeters thick, separated by relatively barren chert layers. And then that interval itself in places is tens to hundreds of meters thick. So this texture, whatever it is, which is just this mix of organic matter and these lensoid things all strung together in these chains, was a major facies at the time of deposition. This would have been covering huge areas of the seabed in this particular basin. And there's a particular kind of acritarch that we extract from these. I'll try and remember to show you later on. But again, in thin section, these look like lensoid structures that show that they were hollow and they were three-dimensional on the seabed at the time of deposition. They're enveloped by the enclosing sediment. So the next set of structures we've got are these braided and quilt-like structures, shown the enlargement there. And these are similar to those pellet chains, and I wonder if these are a taphonomic variant of them, because you often find them together, and these will cover whole slices of the drill core. So yeah, they either form these strings of what looks like braided two or more braided filaments in a helix, or what looks like a quilt-like texture from either the braided filaments or these strange ovoid shapes. And again, I don't know if they are separate things or if they're just variations of the same structure, but they're extremely abundant. Then we have these ovoid structures, and again, I'm just calling them ovoids as a descriptive shape. They're not, they're probably not all the same thing, and they tend to form, again, either as little three-dimensional low relief structures sticking up from a bedding plane. They're enclosed by the sediment, so we know that they were there at deposition. They have a different infilling inside them. Again, usually silica, but there'll be sometimes detrital material in there. Most common ones, these ones on the left-hand side that I've cut in half so you can see inside of it. And then the more complex and weird ones tend to be much rarer, like the one in the middle there, which I think is several different overlapping structures. I think we've only found like 2 or 3 of those ones. We have these lobate structures. And they're kind of similar to the discoidal ones, but instead of having a surface covered in those little, whereas the ovoid ones tend to have a surface covered in with a texture like chainmail, like little plates or tiles. These ones have a more pustular texture to them. And again, they look like they were three-dimensional structures on the seabed that were then draped and covered and buried. Some of them look like they were buried quite quickly because they're at the base of event beds. And these particularly unusual ones on the left-hand side there that have these bifurcating little tubes coming out of them with collars, and those tubes are full of, again, silica, but also carbonaceous spheres that are usually wrapped or coated in phosphate. So yeah, very unusual. These ones pass through several layers of sediment, and it's like something's been pushed over and squashed into the mud by an event bed. And you can see where it's all warped and wrapped around them. This one I've just been calling a lensoid structures, and because in 3D again, it's a large lensoid shape. It looks flat here, but it's convex in multiple directions, and it's made of these tiny little overlapping plates like these you can see in the SEM image there, and they're arranged into long trains, and they tilt at the sides to take in the curvature of the structure, and again they're enveloped by the enclosing sediment. So it was something that was there and getting buried at the time of deposition. And so where the red arrow is, it's pointing to these ridges, and then in between each ridge, there's like a little valley where it's like a corrugated texture. First, I thought that these were slicken sides or slicken lines, but there's no form or shape of deformation around them, and they're full of organic matter, and that's layered within the silica, and that forms these on almost a nanoscopic level. So, there was a sketch at the end there, and it's disappeared. So these ones are, again, I'm calling these dendritic structures, and they cover bedding planes, but they also go up through bedding planes as well, and which I will show you in the next image. And so if you look where the red arrows are, you'll see these kind of little ovoid, tiny little ovoid or elliptical structures. And in that red box, which I've enlarged on the side, you can see that they form these nested chains, and these will branch and bifurcate and cover bedding planes. And they're often associated with these very faint or large ovoid impressions, which are what I was trying to show there with the green dashed lines. These things are really low relief, and they're really, even with using low angle lighting and a macro lens and polarizing lenses, these are really difficult to photograph, even if you can see them really well under the microscope. It's extremely frustrating. So this surface as well is covered entirely in organic matter, and that covers the sediment, which is made of clay and chert again in this particular sample. And then these little lensoid dendritic structures, they're also made of nanoscopically layered organic matter and silica. So this is a cross section there. So if you look at where that blue star is, that's the top of this section here. And so you can see where this whole dendritic structure has gone up through the sediment. But if you look where the red arrows are, you can see there's these extensions that come off the main structure. And what I think is happening here is that whatever it was trying to keep pace with the rate of sedimentation. So not that this was just like a tall thing stood up, it was basically benthic and flat, but it's growing out across the surface, and then it's been buried, and then it's tried to grow above that surface again until it was eventually buried finally by an event bed. And you can see there in the enlargements what these little plates are. And if you look very closely at the large one in the green box, you can see it also looks like it's made of tiny little overlapping plates. And that's what we see in the SEM images as well. And as I said, these are made of organic matter, and they're layered with silica, very fine little flat growths of silica that are interlaid on the nanoscale, really unusual. At first I thought this was some kind of vein, but the more I looked at it, the more I could see that the sediment around it and enclosing it was draping it and piling up on it. There was no cross-cutting, there was no physical destruction and displacement like you would normally see with a vein or like a hydrothermal structure. So it took me a long while to kind of get my head around what I was looking at. So here's what one of those scales looks like, or plates, whatever you want to call them, looks like under an SEM. And it's, again, you can see it's overlapping smaller plates, and they always overlap with the same geometry. These things always have very tightly restricted size ranges, and they're always extremely consistent no matter which sample it is and which drill core and which part of the basin it comes from. And then on the bottom there, you can see that how complex it is on the underside of on the concave side of it. And then we've got this acritarch, which is the organic fossil that we find with these particular kind of structures. So there's the SEM of one of them in situ with the mineral plates around it. And then that's the thing that we extract from the macerations. Then we've got these really large nested structures, which again, they're enveloped by the sediment. You can literally see them folded over on the sediment with the sediment around them, like it was draping something that was there already. They're always at the bottom of event beds, and it's like the event bed has pushed it over or carried it and dropped it down. And they're made of these, again, large plates that are a mixture of organic matter and silica. And the smaller plates seem to grow out from underneath or come out from underneath the larger overlying plate, which you can see in the SEM there with the red arrows. And then they extend towards the point of the structure. And in some of these, when we find them, have this really unusual net-like structure there, which you can see where these arrows, the blue and the green arrows, are pointing to on this particular sample. And they're often associated with their own type of acritarch as well. And there's a really large one just sat on the fossil there. That's what the red arrow is pointing to. And these are the rarest ones at all. We've probably out of all of them, out of hundreds of samples, we've probably only found maybe 10 or so of these. So we went back and had a look at these ooids as well, because there's a subsection of these ooids that have this really peculiar shape that I said looks like a Brussels sprout. And we broke one open where I had a thin section that showed it was full of this organic matter. And we found it's full of these organic filaments in between the layers, but encased in the sediment. And so we checked it with Raman, and it is kerogen, and it is kerogen that is that has experienced the same temperature of diagenesis as the other fossils that we get from this sediment. So it looks like these things aren't some kind of contamination. which is something you've got to be really careful from, because that's what I thought these were originally. If we zoom in on them, you can see there's one of the ones in the thin section, and they're all completely covered in the sediment that was overlying them. They've still got bits of it stuck to them. And if you look at the blue arrows on this one on the right, you can see where it's coming out of the rock and it's been exposed, and then it's getting buried under the interred back into the rock again. So these things are definitely part of the host sediment, but I've never seen anything like them. And there's another one there. You can see it's much darker because it's carbon, and this is iron-rich clay it's next to. And if you zoom very thin, you can just make out these little striations on it, or lines, and they're gaps between what appear to be, again, overlapping little plates or some kind of ornament on the surface. And our EDS data there shows it is made of carbon, and it's got a different composition from the surrounding clay, and the aluminium and silica that's on the filament is actually the stuff that's stuck there. That's those lighter flecks. So that's something that's in this subset of these oncoids and ooids that we had no idea was there. So what I did next was I mapped out where we find these different kinds of microbial structures, and then compared them to the more traditional well-known ones that we found. And we found that they're actually quite restricted in terms of their environment. So you start to get them on the inner shelf. You don't really get them in the shallow areas or in the carbonate producing settings. They're only in the clastic producing settings. And then they become more abundant and better preserved and more diverse as you go offshore. And once you're below Fairweather Wave Basin, then Storm Wave Basin, the really deep end of the basin, these things just absolutely dominate. That's really interesting, because when you compare it to the preservation quality of the acritarchs, you find that preservation quality of acritarchs, abundance and richness of them, like how many different species there are tightly correlated, but they anti-correlate with statistical significance to the abundance and diversity of the microbial structures. So as there's more microbial structures, there's less fossils, and those fossils that are acritarch fossils that are there have worse preservation. So whether this is an environmental effect or whether it's because the microbial structures are literally sealing the sediment, we can't say for sure at the moment, but there's definitely some kind of anti-correlation between the two of them that's really interesting. But in terms of astrobiology, well, we're now thinking about, can show again how rapid orthogenesis and certain types of environments help preserve these microbial structures, if that's what they are. Rapid burial can also help too. And so there's an environmental control on distribution. So if you're trying to look for these things on another planet, you might want to think where in your basin, which is your crater lake, that you're going to look for these things. It's not just the shoreline and the tidal zone, but actually you're getting stuff out in the deeper parts of the basin. And that there's unusual forms. So we might need to think beyond the typical kind of elephant skin and stromatolites that are found in shallow water and carbonate settings. So that was the Proterozoic. And now I'm going to go even further back in time to the Archean, or rather Paleoarchean. This has been a huge international project, and as well as Emmanuel and Yannick there, and Alex, with those a massive, really helpful on-site team, and helping with the drilling, especially the local people like Pumi there and Astrid, who know where to go and what to look for and who to speak to get things done. So this project wouldn't have been anywhere near as successful or really happened without their help. If you've not heard of the Moody's Group or Barberton before, the Barberton Greenstone Belt goes back to about 3.5 billion years old. We're looking at the top of it, the Moody's Group, which is the yellow stuff on this map. And that's possibly Earth's oldest clastic rocks, the first sandstones. And what we're doing is we've got material from drill core that's been unweathered, and it's from this southern part of the Saddleback syncline there. And it's really, as you're about to see, it's really beautifully preserved material. Some of it I've shown it to people and asked them to guess the age, and I can't. They don't believe it's that old. One of the reasons that the Moody's is so well known is because you've got these things, the so-called crinklies, which are interpreted by many as being microbial mats in very shallow water settings. And I think that there's really good evidence to show that they are. Other people have suggested that there are other things like diagenetic structures are metamorphic, but I think the evidence is stacking up that these things are some kind of microbial mat. The other thing that people want to find that we're looking for in the Moody's group are these acritarchs that were found by Javot et al. Back in 2010, 2012. And these are vesicles, so basically spheroids, that are very large compared to the typical bacterial fossils that we're expecting. But they were only found in one small sample set that came from a mine, and it was underground, and we don't know where in the stratigraphy exactly that's come from, and no one's been able to rediscover them yet. So that's one of our primary targets, is we're looking for these things again to try and find them. So it was a big drilling project, had 7 drill cores, and this is an example of some of the material. You've got your crinklies there again in your shallow water sandstone, and you can see it's not the yellow color it was from the weathered surface material, it's grays and greens because there's a lot of volcanic material in there. And then we've got some of this deep water stuff in the middle with lots of interesting wiggly dark bands in there that we're going to have a look at. And then on the end there, that's some unweathered banded iron formation that has an awful lot of this nice green material, as well as the typical red stuff in it as well. So here's just some kind of criteria that we think about when we're trying to decide whether something's a fossil or not, or might be microbial. And it's a lot of petrography basically, and trying to see if it's something that fits within the sedimentary framework, if it's within the sedimentary layers, if it fits with that environment, if it's a kind of environment where you'd find life, and if there are structural features that you can say, yeah, this is the sort of thing that biology would do, and it's not an abiotic process. So the first thing we looked at is these deep water structures with these what look like little microbial mats, and they're chock full of organic filaments, most of which are really fine and very small, but some of them are quite large and robust and appear to have been stuck out the top of the mat into the seabed and have then been bent over and squashed by the overlying sediment as it's got buried. Got some, actually no, I can't tell you that. Some very beautiful images of some of these things, but I can't go into details at the moment. So we've also got a lot of these nice wrinkly bedding planes, and some of them form these pustules, like where the red arrow is up at the top, and they're inside filled with a mix of organic matter and this really unusual, I'm not sure how to describe it. It's like a crumbly or sandy, it's not sand, it's like chert, but it's got a really porous texture that's grown around the organic matter that's there. And it's highly reflective visually, but also it fluoresces really badly as well. So it's making it difficult to get Raman readings from it, because it just gives you a massive fluorescence curve. We also find things that are I'm calling again lobes and quilted textures, because I can't think of better names for them. And I've just tried to map out the areas that you should be looking at on this little rock chip there. What I'm going to do is zoom in as well on here, and the red arrows show the same location in each image. The far right image there is SEM, and that's our backscatter image, and that's showing you the atomic density. And you can see there's lots of this dark organic carbon there that's embedded within the layers of the actual rock itself, within the layers. And then you've got this much lighter material, which is made of clay and silica, and then this bright material there, which is barite and phosphate, I remember for this one. So if we look back at these textures, we can see we've got all of these little pustular textures, like at the bottom left there, and then these unusual lobe-like shapes and So even some of these kind of half ovoid kind of shapes there. And these aren't, I'm only, even though I'm only showing you individual images, these are things that we find over and over and over again. They're repeated, and they're consistent with their morphology. They're not just like random ones that we found. We also have this interesting image at the bottom in the middle there with the orange arrows. And so you've got this dark material, which is a mix of clay and organic matter, but then it's been removed as I've opened a sample, prepped it, and you've got what look like these little pale grey granules. Well, they're not actually granules. They're infilled pores, where the framework for the pores is organic, very fine filaments of organic material. And so that's not something I've seen before. I'm not really sure how to describe it. It's kind of like if a sponge got infilled with silica. Really unusual. Again, we have these ovoid shapes. There's 3 in a row there with this really unusual inclination to each other, but they're also overlapping each other. And they appear to have these kind of ridged borders to them that where the ridges are always pointing away from the center of the ovoid. And these are quite common as well. Got more of these little ovoids. These ones again have ridges and they have these Y-shaped structures in the middle of them. I'm not sure if that's a real structural feature or if it's because they tend to overlap. And what I'm actually seeing is multiple overlapping structures. But again, we have more of these more ridges, and these ridges it's organic matter with clay again and some silica, and inside it's almost pure silica that's inside them. So it looks like something that was sat on the seabed that's then been mineralized internally and then coated by the overlying layers, and then later on by metallic sulfides. Also have these, in between these layers, we have these, they're kind of like, again, they kind of look like these little ridged chains of ridges. And above and below, we've got clay with organic matter, and that's encompassing them both, and these structures here, and folding around them again like it was something that was on the seabed. And so I've looked at that under SEM, and this is a composite image. It's the EDS data overlaid on top of the backscatter, sorry, the secondary electron image. And so that gives us kind of like an order for how these structures, these minerals have come together. So what I think is happening here is you've got this organic and clay mixture of whatever the structure is, and then it gets internally mineralized with the silica and carbonaceous matter. Then the iron clay forms around it, and that's probably chlorite. Well, it's chlorite that was originally maybe illite or kaolinite. It's undergone a diagenesis. And then later on, you've had the metallic sulfides come in and overprint it. But again, we see all of these ridges and overlapping patterns here are consistent between the different examples of these structures, regardless of which sample or drill core you're looking at. That's just another one of those unusual lobe-shaped structures. You can see the ridges on it there moving outwards from the center of this kind of ovoid or elliptical lobe. So last one we're going to look at are these kind of structures. Originally, I thought that these were some kind of cleavage, but then from the tectonic processes. But then the more I looked at it, I realized that they were inside the sedimentary fabric and that they're actually cross-cut by cleavage and veins. And that they're made of these chains or overlapping strings of these structures that are like silica and, again, nanoscopic silica interlayered with organic matter. So I had a look at them on the SEM, and you can see them quite clearly there. And on the left-hand backscatter image, you can see that there's a lot of this dark stuff flecked all over it and within the cracks and inside the cracks, within the layers of the structure. And so if we zoom in on that, you can see it's these small carbonaceous structures, microstructures that we're not really sure on the origin of them. We're currently looking into that at the moment, but their EDS data there shows it's carbon without oxygen or minimal oxygen, and it's completely separate from the silica and the aluminium that make up the rest of the sample. And if you look where the blue arrows are, you can see down the edge of the sample as though you're looking through a cross section of it, and you can see that these things are actually embedded within the primary sedimentary fabric of the actual sample itself. And if you look on the top, you can see, for example, the one where the orange arrow is there. Even that particular, it's not just that one little ovoid structure. It's actually a larger piece of organic matter that goes down and back into the sediment on the top. So yeah, a lot of work still to do. So surprisingly diverse array of putative microbial structures. And it appears that rapid orthogenesis and burial are at work again, which backs up with what we saw earlier and what we kind of know about other types of microbial structures. It lines up with what Foreman and Heubeck have been saying about how the crinklies got preserved and survived metamorphism and tectonism. You've got this surprisingly diverse assemblage of carbonaceous structures, which I wasn't able to show you at the moment because they're still under wraps. And we're still searching for the spheres, and we've got a lot of other analytical work ongoing. So ran over a little bit there, sorry about that. If anyone's got any questions, I don't know if Deirdre's going to be fielding them.
To ask questions, you can either raise your hand or you can also ask the questions inside the chat, as you wish. I don't see.
Well, this is Frances. Can I ask a question?
Sure. Yes. Please do so.
Good afternoon. I don't see where to raise my hand. Excuse me. This is lovely work, Brooke. Thank you. Absolutely fascinating. Yes, I was wondering about some of the filaments that you were showing. This sort of interweaving network filaments in one of your sort of middle slides. And they look very much like fungal hyphae that I've seen in sediments. And fungal hyphae, they're not just subaerial organisms; they're also marine organisms. And yes, I also, with respect to the Moody's material, my first impression was to wonder about the metamorphic overprint and structural strain, etc., on the features that we're showing. But as you say, you managed, you thought about that yourself, and you've been able to show that, or at least convince yourself that this is not the case.
Yeah. So for the first one, that's my first thought for the filaments was fungus, some kind of fungal contaminant. But then when we see them in these, wrapped up in these ooids that were formed on the seabed, like I can't figure out a way how a later contaminant would get in there and look exactly like something that was folded up within a grain. It wouldn't surprise me if there was a way that could do it, because you know what fungus are like. But yeah, and then the fact that it's got a Raman signature that matches the Raman signature of the things that we know are acritarchs. Could it be a fungus that from back then? Well, I don't know. We're still trying to figure that one out. So that's an open question. But if it did turn out to be some kind of contortionist contaminant that somehow got in there and was able to mimic, say, the Raman signature and the petrographic textures of an actual fossil, that would be pretty interesting. I wouldn't be upset about that. It'd be really interesting. As for the Moody stuff, yeah, my background isn't so much paleo as it's from sedimentology and a bit of a closet metamorphic fan as well. So my initial interpretation of most things is that it's going to be abiotic, and that's why I've been trying to have an abiotic first interpretation before I try and move on to anything biotic. But with these things, structures that I'm showing you, they're always cross-cut by things like the overprinted by veins and the cleavage, the prominent cleavage that you get a lot of the time. And that's why I've ended up noticing them. Like a lot of these things, it's just serendipity that I've noticed them, like dropping a sample at the right angle so it catches the light in the right way.
Well, thanks a lot for your explanations. As I said, fascinating work, and congratulations.
Thank you.
Okay. Anybody more? Let's look at the chat if there's something there. Don't think so, but I mean, it can be. Yes, if that was it, I would like to thank our speaker again and stay for 1 minute because our host has something to show to you.